On the cannabis market, every month there's a new strain everyone's talking about. New drops, new crosses, new hype. But what if we're actually losing more than we're discovering? Right now, while the legal market is scaling and optimizing for profit, entire genetic lineages, land races, rare cultivars, unique terpene profiles are quietly disappearing, and almost no one is talking about it. Today, we're stepping inside one of the few places trying to change that, the University of Guelph, a real research lab where cannabis isn't just grown, it's studied, preserved, and reimagined at the genetic level. From tissue culture and extraction science to building a public cannabis gene bank, this is where the future of the plant is being decided. Let's go inside. Big thanks to the Federation of Cannabis Farmers for making this tour possible.
Yeah, so this is the University of Guelph Plant Agriculture Department. We are the Jones Lab. Technically, we're called the Mycophytological Lab because we work on lots of different plants and also fungi. We are really concerned about cannabis specifically, and so we do a lot of cannabis biotechnology research. Most of it is centered around tissue culture research, but we do lots of other things in like flower production, extract production. So my thesis particularly is focused on cannabis biodiversity and the science of extraction and also breeding for extraction. So this is our upstairs lab. So again, we're a research lab, and so most of our work is based on research for publication, and I like to think of us as being a think tank, right? So like this is where we design experiments, where we prepare media. Currently, we actually have Max and some other researchers within our lab have created a new cannabis tissue culture specific media called PHJ media, which we're very excited about. Continuous publications being put out by students and researchers within our lab. Although it's fairly humble, we do a lot of work out of here. It's lots of fun. So we are lucky that we also get to do a bunch of other forms of tissue culture with other plants. And so this is kind of our pet project tissue culture area. We've got some dahlias, we've got some carnivorous plants, and basically when we have projects that we want to do that aren't cannabis, we all put them on this rack. So this is our HPLC that we're working with right now. Currently I'm actually looking at analyzing some extracts. So this is actually some water hash that's being run here, which is pretty neat. So we're looking at a bunch of different peaks. So right here you can see, this is our cannabinoid curve. You've got some cannabinoids, like CBD, THC curve, THCA curve. But what we're looking at here with all these extra peaks is actually really interesting because these are likely minor cannabinoids. Now we don't have the minor cannabinoid standards in the HPLC right now, but we are about to run the standards so that we can quantify what these peaks are. What a lot of producers want to know, and what we also want to know, is what makes certain varieties good for extraction and different forms of extraction. Because there's a big push for solventless extraction within the industry, and that's great. However, there are significant gaps in cultivar diversity that are just not represented within solventless extraction, simply because they just don't have the right type of resin. And so I work a lot with sativas and equatorial varieties that they don't have the right type of trichomes to make really good and efficient solventless extracts, and so they are more tailored for hydrocarbon extracts. The right type of trichome refers to when people talk about washers, like washable trichomes, and so typically those are described as trichomes that have large heads that break off very easily with agitation. And there is a physical component of that just in trichome morphology, that bigger trichomes do seem to be better, but cultivars with bigger trichomes don't necessarily have trichomes that break off more easily, and so you're looking for that trait as well. And then there's also a chemical component that you have some trichomes that just physically burst when you put them in cold water. So typically we're talking about fresh frozen water extraction, which will then go for live rosin production when we talk about solventless extract production. There's also this really interesting concept that when you do flower rosin, so flower rosin, you take just some flour, that dried material, or ideally I actually like material that's just not quite dry and still a little bit wet, and you have a little bit more of a sesquiterpene component to it. And also basically the terpenes there are more terpenes that haven't quite left the plant yet. When you do flower rosin with plants like that, you get to see this effect where it seems like the terpenes are acting as co-solvents, and so there is some additive effects. How generalizable that is for production remains to be seen because flower rosin isn't exactly a scalable process. But it is really interesting as a small batch way of looking through phenotypes for breeding or for research because you have a quick and easy way to actually look at a resin profile. So yeah, a lot of my work involves basically bringing all these concepts together and just trying to figure out exactly what is going on. We're going to go into an area where we will swap out our shoes, and I'll ask you guys not to touch anything. But yeah, we'll go through and just look at everything that we got going down there. It's to have a research license, right? We do have these security requirements, which are fairly strict. So that's why you'll notice too, there's a series of locks, like any LP, right? It's the same kind of idea here. But luckily for us, we are under a research license. And so what that means is we are a little bit more free to just focus on the research and less of the production aspects of things. So to bring plants into culture, basically what you're doing is you're taking cuttings as you would off a mother for clonal propagation, and then you run it through a series of sterilization processes where basically you are surface sterilizing the plant. Typically what's involved is a bleach solution, a 20% bleach solution for about 10 minutes. So you're bleaching the plants. You're then sterilizing everything that's going in here. So your hands, your tools, your vessels, and then you're taking your sterilized plants in there. We chop them up on paper into nice little explants. You can think of them as basically microclones, so to speak. It's actually really remarkable if you've never seen tissue culture before, because the plants just grow with a cut stem right into the media. Eventually they do grow roots, but what's happening is essentially the plant is just taking up the sugar directly from the media, and you get a beautiful little sterilized plant growing, and then you allow that plant to grow up, and then you basically can bring those back into the hood and multiply them. Tissue culture allows us to have a clean plant program where you have copies of your important genetics set aside, growing external to your cultivation area. So if at any point you need to bring out, those cultivars, you can bring them out and you have disease-free, pest-free cuttings. This is our warm chamber, and so this is what I was talking about. This is what it looks like to have cannabis plants in tissue culture. So we have our culture vessels, you have our culture medium, which is the gel that you can see there. And these are at various different stages of growth. So we're actually working on developing a public cannabis gene bank here at the University of Guelph. And so we are really concerned about cannabis biodiversity and preservation around the world and all the trends that are happening with that. And so we are currently trying to bring in as much rare, old, landrace heirloom stock as we can to preserve that for the future. So it's hard to see with some of these bags, but we've got a bunch of equatorial varieties brought in here. We have an old Angola red cutting from the White Buffalo Seed Collective from within Canada. We also have a bunch of really beautiful old haze cultivars, original haze, some NL5 hazes, some of the Dutch hazes, a bunch of other Colombian land races, Thai land races. It's really just about protecting the plants and keeping these plants around, because as times become tougher for producers, we're seeing that a lot here in Canada, that producers, it's tough for people to actually make money with how the weed market is here in Canada at the time being. And so when that happens, a lot of diversity actually gets lost. It often happens that, I'm a plant breeder and I like to produce production strains. You'll make a hybrid that's better for production and you like the traits better than the parents. Keep those parents around. Keep seeds of the parents, keep cuttings of the parents, because it's really easy to get excited about new things. And then eventually 1 year down the line, 5 years down the line, 10 years down the line, you start to realize that what you had was actually just as good, if not better, and that you were just excited about the new thing that you could make. And we see this trend, all over the world. And it's really just an increasing trend because there's this really big desire and need for constantly new varieties, that producers are adding to their menus. So just don't forget about the older varieties and what you made things out of, because that's the backbone of breeding and that's the backbone of, keeping these things around. Plant conservation is not something that one person or one group of people can do. It's something that ideally everyone can do. Even if you just have one male and one female, make seeds, preserve that line, keep cuttings of those plants, because anything you can do to help really helps and it really matters. So people who are feeling disempowered, like home growers, tent growers, people who have a smaller area, you can really still make a big impact by keeping lines and populations around. Just work with what you have, make seeds, and just keep them around and share them and give them to people. We are just doing it because we have the opportunity to do it with our research license here. But ideally, we want to see, cannabis gene banks everywhere, right? Like this is a stronger program if we get these going internationally, all over the world. And so we can do this because this is a team sport. This is not an individual sport. And it's a lot of work, right? It's to try and quantify all of these differences in all the cultivars that we have available. It's mind-blowing, right? Like there's so many plants to be grown out. And what I really want to stress is that this work needs to be done now, right? As we speak right now, we are losing a lot of diversity around the world, even within Canada, like we talked about. And so the urgency of this issue cannot be understated. And so that's why, we're very excited to get going with it. But this is work that needed to happen in the past, and it needs to happen now, and it really needs to happen in the future too. So this is, right here you can see this is regular just nodal shoot culture, which is like standard tissue culture. Then right down here we have some meristem culture, which involves basically excising a very small shoot apical meristem from an ex vitro cannabis plant, and then we put it in petri plates in a different kind of media, and then we grow from meristem. Cold room for culture. Let's just shut that door behind you there, Andrew. So yeah, if you want to take a look at here are some freshly subcultured plants. The reason why we use a cold room is just for longer storage. So one of the benefits of having a tissue culture cold room is we're able to put cultures in this room and they can thrive for a great deal longer than in the warm room. So basically what we're doing is we're using the warm room to propagate the plants until they're of a decent size, and then we'll often throw them in here for long-term storage. And this slows the growth rate down significantly to a time period basically where you can keep boxes in here for about 6 months, and then we bring them out and multiply them regularly. So with tissue culture, there's a lot to be learned, and so essentially the path that we've taken to do that is just constant experimentation on how cannabis responds with different media types, different lighting types, and different variables that you can manipulate within a tissue culture system. There's pros and cons to different medias that people use for cannabis culture. We tend to go for a less is more approach and really just try and keep the plants alive and growing as they are. And don't use too much other plant hormones or things like cytokinins, because there are a lot of issues that are currently being brought up with producers on epigenetic change and somoclonal variation that's happening with plants that are in culture. And so, yeah, there's a lot of ongoing research on exactly what happens due to the stress response of putting cannabis into tissue culture. And so it is, especially bringing plants in, it is a slightly stressful process for them. So typically what we see is that when we bring plants into culture, there's a stress response and some cultivars don't like it. So like anything with cannabis production, it is very cultivar specific. So we do find that some cultivars really like being in culture and grow really well. And other cultivars don't like it and they're a little bit more challenging to work with. So lots to learn with continued research.